Hi everyone, my name is Betty Liu, and today I'll be presenting to you the contents of my Eureka paper, the effect of cytosol-exposed lysine residues on the cytosol membrane distribution and serine 129 phosphorylation of alpha-synuclein. First, let me give you some background. So, what is alpha-synuclein? It is a neuronal protein regulating synaptic vesicles and plays a central role in the pathological cycle of neurodegenerative diseases, including Parkinson's disease, multiple system atrophy, dementia with Lewy bodies, and Alzheimer's disease. Familial Parkinson's disease is associated with alpha-synuclein missense mutations, locus triplication, and increased gene expression. Alpha-synuclein is composed of 140 amino acids, as shown right here. There are three regions: the N-terminus, the non-amyloid beta component, and the C-terminus. The N-terminus forms an alpha helix with 911 residue repeats with consensus core motif KTKEGV, as you can see here. The helical wheel diagram represents the alpha helix of alpha-synuclein, which is in the N-terminus region. It depicts repeats 1 through 7 with numbers as positions. Positions 11, 7, 3, 10, and 6 are hydrophobic and interact with hydrophobic fatty acid chains of membrane phospholipids, which are shown as black lines right here. Positions 4 and 2 contain positively charged lysines, which are these Ks, and they interact with the negatively charged phospholipid head groups. Positions 8, 1, 5, and 9 are hydrophilic and have a higher affinity for the cytosol. Over the years, scientists have strategically designed many variants of alpha-synuclein and investigated how design missense mutations affect the biochemistry of alpha-synuclein. All of these strategically designed variants reveal how naturally occurring Parkinson's disease causing mutations, including A53T, A30P, E46K, H50Q, A30P, of alpha-synuclein affect its conformational homeostasis. For example, the E46K is a naturally occurring mutation, but the P53K mutation, which is a compound mutant composed of E35K, E46K, and E61K simultaneously, extrapolates the effect of the mutation E46K, and it shows significant increases in alpha-synuclein toxicity and membrane binding, as well as vesicle cluster formation. Next, I will tell you about the design of this experiment. First of all, I have designed 4 mutants to extrapolate the effect of E46K and examine how they are different on each of these positions 8, 1, 5, and 9, which are cytosol exposed. So P13K is composed of 3 mutations G31K, S42K, E57K. And all of the other mutations are also composed of 3 mutations which have changed into lysine. The simplified helical wheel diagrams have many representations. The blue lines represent mostly basic positions. The yellow represents mixed hydrophilic positions. Red represents mostly acidic positions. The gray lines are primarily uncharged positions, and the blue dots represent changed amino acids. The purpose of this experiment is to examine the biochemical effect of cytosol-exposed lysine residues on cytosol membrane distribution and serine 129 phosphorylation. I hypothesized that cytosol membrane ratio would decrease compared to wild type because of the increased electrostatic attraction between positively charged lysines and negatively charged phospholipid head groups, which would lead to greater membrane binding. I also hypothesized that serine 129 phosphorylation would increase compared to wild type based on similar work with E46K, which is the naturally occurring mutation. Next, I will move on to the methods and results. First, I will talk about the DNA sequencing analysis. First, I opened the file which was given to us by the sequencing company And I opened the AB1 file, which is composed of all these hill diagrams of the DNA sequencing. And I copied the sequence into the National Library of Medicine's BLASTX database. After pasting the sequence and blasting for results, I copied the results for alpha-synuclein, and this was the result. Basically, I confirmed that the mutants were designed correctly with the DNA sequencing. It showed that the submitted wild-type alpha-synuclein amino acid sequence aligned perfectly with that of alpha-synuclein in the BLASTX database, and for all these mutants, they had exactly 3 deviances in the correct locations. For example, for P13K, it's here, here, and here have been changed into lysines. The second method which I used to analyze was the Western blot data. I used the software Fiji Image J to invert the colors and take measurements with equal area. Then I imported the results into Excel and I subtracted the band means from the background means, calculated the ratios, and normalized them to wild type. I copied and pasted these results into Prism, and I ran the one-way ANOVA and Dunnett's post hoc tests according to the program's guidelines, and I generated graphs from them as well. So after analyzing, I found that the cytosol-membrane ratios of the mutants were lower than that of wild type, as you can see in the bar chart right here. The one-way ANOVA test yielded a p-value of 0.0113, which is 1 degree of significance. The Dunnett's multiple comparisons test showed 2 degrees of significance for P13K compared to wild type, and non-significance for P53K, P83K, and P93K compared to wild type, possibly due to the lack of sufficient data or the high variance. The third result of this experiment is that the serine 129 phosphorylation was consistently higher for all mutants compared to wild type. So, the one-way ANOVA test yielded a significance value of 2 degrees, and for the multiple comparisons test, the p-values were 0.0102 for P13K, 0.0462 for P53K, 0.0271 for P83K, and 0.002 for P93K. So, except for P93K, which had 2 degrees of significance, all of them had 1 degree of significance when compared to wild type. And as you can see here, the phosphorylated serine 129 rates for mutants were always greater than wild type because they were always greater than 1 after normalization. Finally, I will talk about the significance of this experiment. This experiment has 2 main findings. The first is that cytosol-exposed lysine residue increases vesicle membrane binding, and we could see this from the decreased cytosol-membrane ratios of the mutants. This is due to increased electrostatic attraction between positively charged lysine residue and negatively charged phospholipid head groups. The cytosol-exposed lysine residue also increases the phosphorylation rate of serine 129, and this is the second finding of this experiment. Although the exact mechanism for phosphorylation of alpha-synuclein is still unknown, it may be that when alpha-synuclein is bound to membranes, cellular mechanisms phosphorylate it. This experiment also has 3 major contributions. The first is that it highlights the mechanism of pathogenesis for the E46K mutant that causes familial Parkinson's disease. And this is because the substitution of non-basic amino acids for lysine on positions on the cytosolic side of the alpha-synuclein alpha helix results in higher vesicle membrane affinity and serine 129 phosphorylation. This experiment also filled the previous knowledge gap in understanding basic amino acid changes in cytosol-exposed positions and elucidated an essential aspect of the biochemistry of alpha-synuclein. Finally, I hope that this experiment has contributed to a fuller understanding of this crucial protein involved in Parkinson's disease and other neurodegenerative diseases. Thank you for listening, and I hope you enjoyed my presentation.